Hi there.
Welcome to YouTube Microbiology Journal Club, where we nerd out big about all things small.
My name is Danny, and in previous life, I dropped out for my PhD in microbiology at the University of Chicago, where I was infecting skin I grew with MRSA. Nowadays, I'm a fact checker for pharmaceutical advertisers and the president of Biotech Without Borders, a nonprofit based in NYC dedicated to enabling the public with the tools of biotech.
My name is Faz, and in a previous life, I got my PhD in microbiology from Imperial College London, where, amongst other things, I was making flesh eating bacteria glow in the dark. And since then, I've also worked as a research integrity specialist, and these days, I'm an editor for an academic journal.
Every week, we meet to talk about microbiology, and today is our deep dive week where we look at each figure in a paper that we chose last week to learn and criticize the scientific findings. This is a journal club, and we encourage our audience to leave us questions and comments.
And the next week will be our news week, where we survey a bunch of articles and choose an article for the next deep dive. So make sure to subscribe to satisfy your microbiology curiosities.
And you can follow along with the papers that we discuss on either week in our shared Zotero library linked in the doobly-doo below.
And we want to hear from you. So please contact us via our Twitter handles or use the comments. And you can also use the MicroTWGC hashtag, which we'll also keep an eye on. And yeah,
Well,
well,
I had my phone, so we had Twitter.
yeah, yeah. I think without further ado, we should start taking a look at this paper, which is titled, and I practiced highly parallelized droplet cultivation and prioritization of antibiotic producers from natural microbial communities.
A real tongue twister.
Yeah,
Parallelized.
yeah. I've broken it down to each like kind of parallelized
Yeah.
droplet. Yes, I think we're gonna have to say it unnaturally like that, just breaking it down to each syllable.
hmm
So yes,
Yeah. So this is I think we mentioned it when we chose it. Like this is almost very similar to the paper that we did at the beginning of this year, which was sort of screening through large combinatorial chemical libraries to find antibiotics. And so this is actually from a background slide from that first one that we did. If you remember, there's giant libraries, millions of compounds, robots manipulate them, and the challenge is somewhere in that library there might be an antibiotic. But what's interesting to remember is that in these libraries, they may be all synthetic, but they could also be based on natural products, which is actually the place where we've seen more antibiotics come from.
yeah. So in the history of antibiotic development, we mostly steal the ideas from bacteria that live in the soil, and
Yeah, this genus Actinomycetes,
mm,
um, which I think like for people who are deep in the bacterial nomenclature, like you can tell they thought it was a fungi before
yes.
they realized it was a bacteria.
Well, yeah, that's the thing about because a lot of these antibiotics come from almost colonial bacteria that think they're fungi. So streptomycetes, actinomycetes, and yeah, and it's almost like a community weapon to make sure that they as like a multi-organism can outcompete other bacteria. So they have these generic products. So these are weapons that bacteria use in battles against other bacteria that we've taken and used against them. So uh,
Yeah, so I mean, I think I'm not sure if we talked about it last time, but antibiotics, there's like a golden age of antibiotics.
yes.
Where they say it's the 1950s, 1960s, and you can see all that list of all these antibiotics there, and then they colored them by where they came from. And
Mm.
they're actually the golden age has a bunch of synthetics, but they have like the two synthetics that have stars beside them. They're actually they're influenced by natural products.
Mm.
So even in the synthetics that we've made, sometimes we get like these clues from or like compounds that we find in nature.
Yeah, and that was like tipping on what we were talking about the deep learning paper that we did earlier this year, where they were looking at kind of organic compounds that have antibiotic properties, and then using computers to build up an idea of other things that we could do to make similar compounds that could also be antibiotics. Um,
hmm hmm and yeah, so if all that diversity, so there's sort of the chemical synthetic diversity we get, some of it's even inspired by the natural world. I guess the place that is very natural to say that there could be more antibiotics in is in all those other bacteria out there. Right?
yeah, and I think we're going to be hitting on like a kind of a big so in historically, so if you've been in a microbiology course, you've probably done this streaking plate experiment where you take a sample of bacteria, streak it onto a plate to isolate it. And this has been used for quite a while, but the thing is, it's not perfect, and there are a lot of things that don't grow on plates, and we call them unculturable bacteria. So bacteria that we don't know about, because the moment we put it on a plate, they either start to die or they get outcompeted by bacteria that can grow on a plate. So it's almost like this bacterial dark matter that could have so many antibiotics, so many interesting biological products, but we never get to see them because we can't isolate them through normal techniques. So
Yeah.
yeah, over the last couple of years, there's been loads of new techniques that people have been developing to specifically try to uh, find out about these bacteria. So this, Danny found this amazing like picture of all these different
Mhm.
techniques. Uh,
Yeah, it's a
um,
good paper. I think it is specifically talking about like the unculturable bacteria out there. We know about them because of sequencing, right? So
right,
this picture on the left hand side, right? That's
yeah,
a, I mean, it's a phylogenetic tree if you zoom right, if you look right in the center of that,
yeah,
and that's from sequencing. So right, like maybe people are familiar with the fact that we can sequence a lot right now, like even
yeah,
environmental samples you can sequence. Um, and we know that there are tons of different bacteria out there just by their sequences. But the numbers in the white circles, that's like how many we've actually sequenced from each of these groups. And you can see that there are some like four-digit numbers in there.
yeah,
So, it's not that we haven't cultured a lot of things, we have, but they're clustered, right? They're in really specific zones. And that's partially because we do things through plates all the time.
yeah.
And there's like standard medias people use and like conditions. And like that's just limited the scope of what bacteria actually we work with in the lab.
Yeah, I mean, I guess the analogy is you're trying to keep a wild animal in captivity. You have to understand how it works, but we've only been using one specific type of like way of capturing bacteria, and a lot of them don't survive. So the ones that do survive, we know a lot about, but
Yeah.
as you can see from here, they only represent a tiny amount of what is out there. And
Yeah.
so developing these new culture techniques are a way to try and make things more like the wild for these bacteria.
Right.
More, yeah,
Yeah, no,
making
I want to run with that analogy. I think it's great. It's like it's the zoo analogy, right? It's
yeah,
like in the lab we make habitats for bacteria, and we've been giving them the same old like playpen, right? With like the same sugars, the same amino acids, right? Similar temperature, and we say what will grow, and we found tons of things. There's tons of things that will grow in that environment, but that's not the diversity of environments that exists out there in the world. And so, getting closer to that and like iterating through like all these different environments, that's a big part of tapping into this dark matter. And people microbial dark matter.
yeah. And one of the things that was done previously, there's a new antibiotic called Taxonomy, which is one of
Mm-hmm.
the few antibiotics that have been discovered in the last like 10 years.
Yep.
And essentially, what they've done is they basically found a way to culture a bacteria in their own kind of environment. Am I
Yeah.
right? Right? Yeah.
I think it's an awesome. I was very excited about this tech. So that's like the hand-drawn image at the top left.
Yeah.
Is essentially this eye chip setup where they have tiny little holes drilled into like I think it's a piece of metal,
Yeah.
and they dilute a sample of bacteria over it from the environment. So your soil bacteria, you dilute the soil until there's like maybe there's a very dilute amount of bacteria, and when the bacteria, when that liquid goes to each of the holes, in general, only one bacteria will fit into every hole
Right.
by dilution, right? So they dilute it out. So like each of these is its own little container, and then they seal the holes up and with a membrane that's permeable to the environment but impermeable to the bacteria, and then they put it in the environment of choice. In this case, they did soil.
Yeah.
And so that is the iChip technology, and it worked really well. They were able to find different bacteria, screen through them, and find a different antibiotic. And like what I love about this technology is the picture right below it.
Oh gosh.
It-
Yeah. The
yeah, the
what is
denture
that?
put in,
Oh.
or it's like not a denture, but it's a mold of the top of your mouth, right? So
Yeah. Like a retainer. Like
yeah, exactly. And so this technique, like, it's not just for finding things in the soil or like whatever. Like, we are also an environment to grow bacteria, and if we want to understand our own biology and the bacteria that grow in us, we also have to grow some things that we haven't been able to grow before. And the way they did it was they just embedded that same chip into this thing in your mouth, so now you can grow mouth bacteria that previously haven't been grown.
Yeah, and so I mean, I think it's important to think about this not just for the fact that we'll find new antibiotics, but also just have a greater understanding of how bacteria work and what's out there, because there could be
Hmm.
other things that would be really important for us to understand that we aren't getting because we are just the way we're because petri dishes have been around for like 100 years,
Yeah.
and it and feel like maybe there is about time to change that up. And things like technologies like this are ways of changing that up so that we can investigate more and understand more.
Yeah, absolutely. Absolutely.
So, yes, and microfluidics. So this is a big deal now. So there's been a big like kind of advances in miniaturization and
Yeah.
actually
And
controlling.
we've talked about things that use this technology
Yeah,
before, right? We've talked about single cell sequencing, right? Actually, that is you have to be able to separate out single cells, and to do that, they use microfluidics.
right. Yeah. Uh, I mean, we've mostly talked about it in terms of like genes, but they're thinking a bit bigger, but still at the
Mhm.
nanoscale. And yeah, this is a really fascinating way to think because instead of like running one petri dish with a mix of bacteria, what the idea is to have like 100 tiny little petri dishes that are droplets growing
Yeah.
individual bacteria, which
Hmm.
is an interesting because then you've got a situation where no bacteria are left behind essentially, uh,
Yep.
which
If you can fit into a droplet, then you'll have your own chance at growing.
yeah, and also in terms of like the way to analyze these droplets as well means that you are looking at a single bacteria. So if you've got a specific mutant, you're not going to miss it. You're going still going to be able to find it amongst a million of I mean, there are this like scatter graph shows like millions and millions of individual bacteria, right?
Yes.
Yeah,
And the other thing that I mentioned here is that side scatter, forward scatter, those are the tools of flow cytometry. So like
yes,
it's really out of flow is that we're starting to get at this. It's like we have ability to we have ability to shoot lasers through things and see what light comes out. We have the ability to manipulate small droplets. Those are the types of technologies that are building up to this. And people in the culture, I think some people call it culturomics.
yeah.
Like in the culturomics field, like are utilizing these tools in order to grow things that haven't been grown before.
Yeah, I mean, I feel like that's because like omics indicates like a totality of like understanding everything. I mean, it came from
Hmm.
genomics, and now it's transcriptomics, and now it's going to
Yeah,
apply to everything now.
anything that like technology allows you to see the overview of it, like without having to, I guess, like manually go in and check everything. It's like something like that. That's the omics movement, I guess.
Yeah, the way to also like handling massive amounts of data all
Hmm.
in one big go, and then find a way
Yep.
to pull stuff out of that. It's
Yeah,
so this is almost as much an advance in data science as it is in technology. Uh,
totally. Yeah, I think both things had to go hand in hand in order to be able to deal with these types of problems. So very quickly on the left-hand side, that's like one of the early, that's the 2002 paper. So that's like kind of earlier, right? It's very flow cytometry based. I think they
yeah,
even flow cytometers to do it, right? Like they were using commercial things that were out there already,
yeah.
right? Put cells in droplets and measure like, oh wow, like can they grow in this thing? What do they look like? How do we sort through them to find the ones that like are more alive, that are clumped, right? Like we have to find all those droplets the ones we actually want to deal with.
And this side is basically 18 years later, and we've
Yep.
advanced a lot more in terms of like types of lasers we can use to focus in on these droplets to check what's fluorescing and get more detail about what's in the cultures. And
Yes, I think another big part of what has been accomplished since then is that we're able to just like deal with droplets in bulk. Like
yes,
there are ways of like manipulating droplets. So like if you see B, or actually any of those panels, A and B,
mm,
like all those droplets, they're all separated, but they're together, right?
yeah,
Like they're essentially in a single mixture and they're all separated. It's oil that it's used to separate them. I think we'll see a little bit more of that in the next slide.
yes, yeah. I mean, I like the hexagons and bestagons in this section here. But and you can see all
Yeah.
the yeah. And you can see like different bacteria, like kind of these are flexible. Some of them are flexible, like kind of spindly little chains, and some of them are more like dots. So
Mhm.
it's kind of really interesting to see that at this tiny level. But yeah, next slide. Sorry, I got microfluidic techniques. Uh,
Yeah, so this is how you get those images right on the side. This is how you make the droplets. There's kind of a scale. There's a scale NB, right? Like you can see it's not big, 9,000 microns, right?
yeah,
So these are
that's
tiny. That's 9 millimeters.
9 millimeters. Yeah, so yeah, that's pretty tiny. But I mean, I think people can understand that on a scale that because you
Yeah,
can see you can imagine how big 9 millimeters is. I imagine, and then on the actual droplet itself is about like 4 millimeters. And then inside there,
yeah,
and that's what's used to. This is the mechanism for actually generating the droplets, though. So
yes.
the droplets themselves are really tiny.
Yeah, the droplets themselves are very tiny and they're made in these very tiny constructions. And so a big part of that is that, right, like oil and water don't mix, right?
Mm,
So like that they're using oil to separate out these droplets. And then because they're so small and the surface tension, I guess, just keeps them together, they add some other stuff usually into the oil and
yeah,
it just lets these like little droplets like float around. They're essentially in a emulsion. They're like a vinaigrette.
yeah, yeah,
Actually that's a really good description of them. And except in a vinaigrette, like because you're beating it together, the droplets will be different sizes, but they have a controlled way of making these droplets. Like you can see the cells flow, and then the oil like pinches off.
yeah,
Uh, um, Constant sizes of the cell mixture, and yeah, and then you end up getting cells into them. And if you control the dilution of the cells, then you can have a dilution so that you're guaranteed to get single, or like you're more likely to get single cells. But of course, it means you're also going to get empty droplets as well. But that's like part of the technique.
right, yeah, yeah. And I mean, I'm just like checking to see whether like my cursor's, but this is looks pretty cool. Next slide. Ooh, yeah, antibiotic producing microbes using these droplets to culture
Yeah, so
them.
paper isn't number like this paper isn't the first time they've considered it, right? Like actually the same
Mm,
authors published already, right? That this would be a
right.
really interesting thing to do. They didn't actually search for unique antibiotics or anything like that, right? Like they're
Mm,
primarily I think they're very concerned with the technology, right? Like, how do you set up
yeah.
this type of system, right? How do you go and look at stuff? And so they had already done an experiment where they cultivated things in tiny droplets and then they injected reporter strains into them and saw that in when they did it with communities of bacteria that made antibiotics, like they could show that that fluorescence went down. So that really, this whole paper is just an extension, right, of work that I guess, this group has been doing for some time now.
Yeah, and I think it's quite important that they. I mean, it's understandable that they actually try to make this as public as early as possible, so that other people could understand and benefit from it. Uh, and you can sort of, and it's quite interesting what they because they have a fluorescent reporter strain that they inject into the little droplets of the other bacteria. So there are two types of bacteria in this. One's a reporter, one's the bacteria that they sample. And if that bacteria they sample has an antibiotic, it kills off the reporter strain,
Yeah,
and so it changes color of the droplets,
yeah.
which we can kind of see here,
Yeah,
where,
I
where
think they're demonstrating it. I think each of those is like a different fluorescent marker that they're using. I think
yeah,
I don't remember what they were actually, just off the top of my head, but I know one of them is for the fluorescent bacteria. One of them is for empty bubbles, right? They had some way of
yep,
marking emptiness. And then one of them is, I guess, just for oh yeah, three is definitely the reporters, I think. Um,
yes,
and yeah, and in an image where you composite them all together, you can see these droplets exist all together, but they're different colors, and they're able to actually sort out the ones that they want and the ones that they don't.
yeah. So that brings us to Figure 1A, which is the start of this paper, where they kind of show their workflow here.
Yes. Yeah. So, I mean, we sort of saw all the building blocks to this, right? Like they first start by making cold extracted soil extract. So,
Yes,
basically, cold brew soil, I guess is the way to think about
yeah,
that. Make
yeah,
some soil. Yeah. Go ahead.
yeah. No, I looked through it, and yeah, they basically take soil and kind of they eviscerate it. They kind of like blend it. They dry it out a lot. So they put it in an oven at 37 degrees to dry out a lot of the stuff, and then they kind of then filter it and then take out this pure substance this has a mix of all the stuff that's in soil
Mm,
that the microbes live on. which is kind of what you want. You want to replicate the microbes environment as well as possible, but you also want to make sure that it's sterile as well, because you don't want to introduce another bacteria that would kind of interfere with things. And you want to make it kind of stay standardized as well, so that everyone gets the same thing.
So is that?
Uh,
I don't think that's not the cold extracted though soil extract, is it? Because like they have
oh
that's
no,
like
you're right.
the media maybe that they that's the media preparation for all of this.
Oh right, yeah.
Let me just let me drop into
I mean,
this.
yeah, I think I got confused because they actually isolated the bacteria that way. So when they isolated the bacteria, they took a soil sample, they shook it a lot, very hard in the sonicator to shake
Mhm.
the bacteria off the soil particles. Then they dried it out at 37 degrees C, and then they filtered out like half of it. So they filtered out like some of it. So they had a little pause where they pushed the filter through to make sure that only bacteria could fit through.
Mhm.
Uh, and then they had another one where they had like a kind of a solution of all of it, and kind of waited for all the kind of particulates to sediment out, and they
Yeah.
took a little bit of that and mixed it in with the rest of the bacteria.
Yeah. Yeah.
Um,
So it's not it's not sterile, right? Like because
yeah,
yeah.
the bacterial samples aren't sterile because they
Mhm.
needed the bacteria. The soil extract is was
Is
sterilized
yeah, and
to
that's
make.
through and the sterilization is done through filtering, but the extraction has a dry step in it.
Yes.
Yeah. Yeah. Yeah, so there's two things, right? They need the nutrients that the bacteria are going to grow on, but then they also need the bacteria themselves. And
Mm,
the strategy to get the bacteria, they also have to break up, right? Because soil is a bunch of little particles, and the bacteria
yeah.
will also be stuck together. So that's where the drying step comes in to like break up all the little pieces of soil, I guess, to make sure that they only get single bacteria.
Yeah.
Um, and they speak to this that like even though they did all these processes, there's still like a chance. I think it's actually a really high chance, right? That some of these droplets contain more than one bacteria just because of all these like because things are so sticky.
Yes. So they basically tried to made it so that the ratio of like empty droplets to occupied droplets was like there are 30% occupied droplets to empty droplets.
Yep.
So that mean that on average they'd have one cell per droplet.
Hmm.
But there would still be a probability of them having maybe two cells per droplet,
Yep.
and that's kind of unavoidable.
Yeah.
Um,
And then all those droplets again, they can be pooled together. So they have 9 times 10 to the 6 of them. That's a whole bunch of droplets. They all go into a single container, and in that container they get incubated. And
yeah,
containers mostly oil, but right, the droplets remain in that oil, and presumably uh, only things that I guess really like oil can diffuse out of there. But it's so dilute they shouldn't diffuse into the other droplets. So that's like a key part I think about their setup is that they presume that these droplets are independent, right, in this mixture.
mm,
Yeah, and then I guess and then we keep going, but like maybe yeah, we can probably move to the next slide.
yes. So now we've got look at the different controls. So there's you have a negative control, which is just empty. Positive control, where they use streptomyces, which is an antibiotic producing bacterium.
And
Um,
this group of streptomyces, this is the same thing that they did in their 2018 paper, right? So they're essentially taking the work they did before and say this was a really good positive negative control, but now let's put in the unknown. Let's put in the soil
mm,
community and see if we can find something.
yeah, and I mean, looking at those microscope, sorry, those images, you can see that the soil community is very diverse. You are seeing lots of different structures in those microcolonies growing in there.
Yeah, totally. And it's 28 days later is what they do, and I think they incubate at 20 degrees Celsius, so it's pretty cool because the soil is a fairly cool place.
Yeah, I mean, I guess it's kind of cool from the perspective of most incubators, which are generally kept at 37. But
Mhm.
soil, it can be quite depending on where you are actually. So if it was, to be fair, I can't remember what country this was. I think this was from Germany, so they
Yeah.
might just have very warm weather there for soil. But yeah,
I mean,
uh,
these are all like, and I think, right, we talked about like people trying to cultivate things that haven't been cultivated before, right? These are the types of things that you encounter when you're trying to do things in the lab, right? Like,
yeah,
it's not like the soil, because if you think about soil outside, actually there's a gradient of temperature, right? There's stuff at the top all the way to the bottom. So like, every time you fix the environment, right, you're going to favor certain growth over other growth. And the whole
Yeah.
point of this procedure is trying to just push it to something else, push it to what we think would grow in soil. And that is also amenable, though, to our own equipment. Because you can imagine at different temperatures, I'm sure this oil behaves differently, right? The
Yeah.
particles behave differently. They won't be able to necessarily get a huge range, but this is what they're going with for our purposes here.
Right. And that brings us to the other. So in order to show whether the droplets work, they mixed in they compared it to traditional agar plating techniques. Uh, so this is kind of, if you've ever worked in a lab, you've probably done this kind of traditional plating technique where you put a droplet of bacteria on a petri dish and you kind of spread it around using a spreader, and
Yeah.
then leave it to incubate and then see what grows afterwards.
Yep. And so they did that with yeah, they did that with their soil samples. They put it through this droplet way, and then they put it through the plate way, and they collect it all at the end, and then they see how many or how many colonies grow, right? Or how many droplets have something growing in them. And from that, they can estimate, right? Well, I mean, on the plates, you can count them, right? You can count all the dots
Right.
you see. But in the droplets, they have to sort of count every droplet that they see grew up a little bit. And I think
Yeah.
she ended up maybe estimate. I think it's an estimate of some sort, where they count like they sample a whole bunch, right? And they say the occupancy was this much, and then we have this many drops. So then this is the CFU of that particular way of doing it.
I mean, we do the same with bacterial spreading by diluting, because sometimes you get a bacterial lawn, uh,
Mm-hmm.
which basically where the bacteria just form this flat kind of layer, and it's impossible to count them. So we'd have to like dilute it down further until we can get a plate that's actually countable.
Yep.
Yeah, I guess so. It's understandable that that would be a similar thing to the droplets. Might not have the chance to count all 9 million cells that they've got in there.
Exactly. They just count a fraction of it, and then you figure out from the dilution factor where it's at.
Yeah,
So from that, they can show that this their droplet technique does seem to produce a lot more growing single cell bubbles, right? Um, it's
yeah.
able to isolate these colony forming units, is what we call them when they're on plates. Yeah, and I guess in droplets they kind of call them colony forming units too here.
Yeah, and and with the plates, plates become more accurate overall for when you dilute them more, because then you can get more counts. But obviously, the problem with that is that you lose a lot of rare bacteria that aren't very well present. And if they're not present in a certain like kind of concentration, then you're going to lose them once you dilute things.
Yeah, yeah. And then I guess the other thing, oh, I guess we'll move on, but like they take these two samples and they're going to do some tests on them to show us the differences between the two. um,
Yes.
so our favorite test for figuring out like what the composition is would be 16S rRNA. So that is a gene that's present in all of bacteria. And there's a lot of research that's been done to help us categorize all bacteria based on short sequences of that gene. And so here they did that. They take those two samples now and they get all the DNA out. It's a mixed composition of DNA, right? There's DNA from
Yep.
all the different bacteria that are there. And then they sequence it and then they compare it to a big database that says this small sequence means this type of bacteria. uh,
Yeah.
and they create these huge data sets, so big that, of course, we have to use principal concordant analysis in order to decompose the complexity.
Yeah, get an AI to kind of flatten the data so that we can see how related things are to each other. At the very top we see the soil and extracted soil community. So these are the from samples that were taken from the soil and from samples taken at the end of the extraction process.
Mm-hmm.
So you can see that from that they basically say that, oh, the extraction process, we don't lose any diversity in the bacteria. So
Actually, let me just clarify for this first one. So like after they get the table of everything that's there, then they make a phylogeny from it.
mm,
Right? So they say how are all these things related together, and then it's
mm,
from those. This is actually a comparison of trees, right? It's like
right.
because it's trying to say like like if there's like a group of bacteria that don't appear in the other group, then the distance gets bigger.
Yes.
So that's what we're seeing on this principal component analysis. It's actually a comparison of trees, and that is considered it's considered a measure of diversity. Yeah, as you were saying, diversity.
Yeah. Yeah. And yeah, well, we can, and like in the middle, we've got the droplet samples.
Hmm.
Right at the bottom, we've got the plate samples, and we've got the positive controls kind of a little bit further off because positive controls are just basically one type of bacteria.
Yeah,
So those are going to be very far off for everything.
yeah.
Um,
Five types, five bacteria. That's like the known, the known
yeah,
five types of bacteria. But yeah, the from this type of analysis, you can see that the plates and the soil seem to be quite different. Or sorry, the plates and the droplet cultivation techniques, they grow up different things. Uh,
yeah, and I think the distance as well is quite an important thing because the fact that the droplets are quite are closer relative to the plate samples is also quite an important thing to note.
mm, yeah, like that. The cluster of droplets is closer to the cluster of soil than the cluster of plate samples is to the cluster of soil, right?
Mm, yeah.
um, And another way to show this would just be to go through that big table and say what phyla do they come from. So phyla, right? In taxonomy, you basically zoom in to the species, but there's groups that radiate outwards from that all the way up to phyla. Phyla is one of the biggest groups. And
Mm,
so in this big group of like this sort of broad strokes of classification, you can already see that, well, of course, the controls, the positive controls, they're just one thing. They're five things actually,
yeah.
but they're all from the same group. So that's going to be all one color. But in the soil, there's a whole bunch of different things going on in there, right? And
Yeah.
the extracted community is very similar to the soil, so that's a good control to say that their extraction is sort of mirrored, right? Like they're not changing the diversity structure so much. They may be destroying all of the types of bacteria in every group that would be destroyed by that methodology that they use, but it's not focused on one type, right? So like
Hmm.
diversity structure remains similar. And then the big comparison you want to look at is the droplets, right, against the soil and the plates against the soil, or even the droplets and versus the plates, right? Those are the two ways we could
Yes.
look at it. And you can already see from this, there's sort of some patterns that are occurring, right? That the droplets have the actinobacteria more represented, which is actually more like soil.
Yeah.
Mhm.
And yeah, you get more like gram-positive being taken from plate samples. So firmicutes are the gram-positive bacteria. You've got like proteobacteria as well. They're very important. So yeah,
But
it's interesting.
proteobacteria seem to be enriched like across the board, right?
Yeah.
Versus soil. Like soil doesn't have a lot of proteobacteria, but like they grow in the lab really well.
Yeah. Uh, and
And
the,
proteobacteria, I think that's where E. coli is, right?
I believe so. Yes. We go previous slide. Sorry. Uh, yeah. I mean, sorry. Is there anything else we want to think about for this one? Or
No, I think I'm good.
okay.
Let's move on.
Okay, so now they use a whole bunch of different diversity measures. These
Yeah.
are various tools that are used by ecology and uh, even like mathematics, and it's. So I did
Yeah.
try to learn about what these were, and I kind of failed, but I can try and.
Well, I think an important summary about these six or these eight different metrics that they use is that it's different from the tree method of
Mm,
looking at things between, right, focusing on the differences between the groups, right, and saying like that drives the diversity. Here they're actually looking at like how rare inside of a group can, like how many rare things can you find inside of a group, right? So like how
yes.
long is the tail in some ways, right, of all the different things. And you get that from A, right? Just number of OTUs. So I
Yeah.
we probably should explain OTUs. Essentially, that's the species definition for bacteria, because it's too difficult to sort of use traditional species definitions on bacteria. Instead, they just sort of set a percentage cutoff of similarity in 16S rRNA, and that percentage cutoff is the species or is the
Yes.
OTU.
So
Uh,
operational taxonomic units is
yeah,
the is a
the most
word.
mathematically defined way of organizing bacteria, but I
Yeah,
think what might be interesting for people to know, they may not know already, is that there's a lot of diversity in bacteria. There's just like so much going on there that it sort of defies the way in which that we would easily categorize things, right? And they're also
yeah,
all like lumps.
yeah.
So we don't have a lot of traditional markers to go off of in terms of organizing the groups.
Yeah, and we have to go by like genetics, which is difficult because bacteria do swap genes quite often, and that makes it, and through phages or through plasmids, it makes it quite difficult to figure out like kind of the genealogy of bacteria because there is so much noise there.
Yeah,
Um,
so they everything gets anchored to 16S. That's sort of how
yes,
the scientific community has collapsed down to make sense of all of this diversity. Is say, let's just anchor it on one thing that we know is on everything, and that we're fairly certain gives us discrimination between different types of bacteria. And then they choose an arbitrary cutoff, and that makes the OTU. So
yes,
yeah. it
and another, sorry, uh,
I want to say in all of these analyses, keep that in mind that like that's in the background of everything that we've made these assumptions about what distinguishes bacteria from each other.
yeah, and the other thing to think about is for singletons are basically a read where the sequence is present only once in the sample, so it's a unique sequence. So if you spot that, then you've spotted a rare variant of bacteria.
Yeah.
And based on that, so you firstly can say, okay, the number of that can speak to the diversity of our sample, but also we can use that to estimate the coverage, how well the sample represents the rest of the community. And those are basically those are essentially what define the next two indices, like
Mm-hmm.
where these are indices used by ecologists to when they're sampling diversity to figure out how well their sample actually represents the community. And
Yeah.
if it's a good sample, then in this case, are you saying that if you sample represents community a lot, then it means that there isn't much diversity in there because you wouldn't expect it to. So
So,
that's
so the thing about this, remember, the thing about these metrics as opposed to the one that we saw first is that these ones are all comparisons into themselves. So
mm,
they're when you say community, community means everything that was read by the machine in some ways.
yes,
In some ways, everything is singletons. To me, that's how I think of all of this
mm,
stuff. It all sort of relates back to singletons. Like, how many unique different things did you find in the list of all the things? And probabilistically, is that reaching like what you would expect as saturation of all the unique things you could find, right? Like one of the ways that people show some of this is like where they subsample from the data set over and over again, and
mm,
they see when do singletons, how many singletons do you find on every subsample, and you can imagine that as you continue sampling more and more, eventually the singletons they kind of they plateau out, right? And then
mm.
that's how you know, like, okay, I've sampled enough, I've read enough of everything there, because the more I read, I'm getting diminishing returns on all the rare stuff that I'm getting back. And these measures sort of all relate to that, and that's
Yeah,
called alpha diversity.
yes. So I guess the main takeaway is that the drop cultivation tends to create a more diversity than with plate cultivation, but not
Yes.
necessarily as much as the soil and extracted soil.
Yep. And in two measures of diversity, right? We saw in the previous slide, right? The diversity was we have more things, right? More things
Yes.
that are similar to soil than not similar to soil. And then here, the type of diversity we're saying is we just have like more rare things in general, right? Like you're finding more unique reads. So there's something about this method that's just helping us find things that, yeah, we wouldn't normally find.
Oh, and now they drill in a lot more deeper into each kind of each genera to see and
Yeah,
comparing.
yeah,
Yeah.
yeah. So like, I don't know what was it like. King something green plays chess on fine green sand, something like that. You remember this?
Yeah.
Wait, there's taxonomy is like right. It's just like circles getting bigger and bigger. The most specific is species. The next is genera. Right? You have family, class, order, phyla, and each group is
Yes.
getting bigger and bigger. So because they have all this data, they have all these things that get associated, you can re-perform the same analysis essentially at every level, right? You can say, what are the top 10 most abundant, in this case, genera, but then they're going to go and they're going to do it for everyone as well. But genera is really useful because it's not as specific as species, right? It's in between, right? It's
Yeah,
the perfect. The reason why we often use this taxonomic classifier is because it fits nicely in like in an in between ground, right? Between species, which is like there's too many of those to really care about, right?
yeah,
Well, those are all the reeds actually. This is the level right above reeds because species is down there at reeds.
yeah. And when you
Um,
start talking about species and bacteria, often you end up using the phrase pseudo species because there can be so much diversity in what we classify as a species that it gets
yeah,
very complicated. Uh, so genera makes things a lot more simpler to understand at this level. Um,
yeah. And like we have some genera that you might recognize, right? They have bacillus here, right? So
yep,
bacillus, that's like gram-positive rod, right? It makes
yeah.
spores. Um,
You may know this from such diseases as anthrax and what
and
the
such foods as kombucha
yes,
and natto, right? Like these bacteria, like even in a genera, there's like huge amounts of diversity, right? But at least it has some like common threads that we can sort of tie to it physiologically, and then yeah, and phenotypically. Oh my God, it's all over the place.
yes,
um, but so anyway, so that's just a quick overview of like why genera is
hmm
chosen. And it ends up being quite informative because you can rank the top 10 most abundant, and the top 10 most abundant, it takes care of 66% of all the
mm,
reads, right? So you get a sense already of the type of diversity that we're seeing, right? Like there's a long tail.
mm,
They always talk about this in microbial diversity. There's a long tail in microbial diversity, right? Lots of things kind of like go out there. All sorts of rare events happen over and over again, right? Continues as you sample more and more. But there still is like a huge amount that's explained, usually just by the first chunk,
yes,
uh, most abundant things. And by zooming into this, we can see a more detailed view of what's different between the plate cultivation method and the droplet cultivation method. And so you can look at the Cohen's d bars to really quickly see that, right? Where the diversity is in favor of the plates, you get the purple bars, and where the diversity is in favor of the droplets, you get the green bars. And so there are in this top 10 list of things that characterize 66% of all the reads. They point out that bacillus and penibacillus and phylobacterium, those are hugely overrepresented in the plate samples. But things like Acinetobacter, Agromyces, and Micromonospora, these are things that are represented in the droplet samples.
yes,
And then again, this is like for people who know bacteria. If you're familiar with the groups, then you can start saying things about the groups at this level, right? Like in some ways, this is the top 30 now that you're
yep,
showing, right? It's like even more than top 10. And top 30, you can see when you do top 30, it covers even more. It covers 90% of all the reads at top 30, right? So that gives you a sense again of that like diminishing returns thing. I want to go back to the Venn diagram though, real
okay. Let's
quick.
go back to the Venn diagram. Okay, right. So
And I think the most important thing to pull away from these types of graphs is like it's helping us understand what's unique, right, in the plates versus what's unique in the droplets.
yes,
So in the plates, there are unique things, but there are not as many unique things as in the droplets. And that's true both with genera and when you look right at the OTU level.
yeah, and I think that's very important from what we want because we want more diversity. So looking at this, you look at which one will give you the more unique diversity, and then you see okay, well droplets clearly because 983 is a much bigger number than 27. I don't think you need to be a scientist to realize that. So I think uh, it's an important.
Yeah. Yeah. So I think uniqueness is key here because like there's many ways of saying what makes things more
Mm,
diverse, right? Because like in this top 10 list, right, you can already see that uh, there's a majority of genera that are actually unique to the
mm,
plates, right? Or are more enriched in the plates. So like that could be a form of diversity as well.
yeah,
And so like yeah, right, like there's many ways of I think that's the biggest one of the the devil in the details things about microbial studies like this is that diversity is a very broad term, and really scientists want to know very specifically what that diversity looks like, and this is helping us get there, right, by zooming in different ways.
Yeah, I mean, I'll talk about this a bit more later, but I think it's not so because with this paper, it's not so much capturing all the diversity, but selecting which diversity you look at. Uh,
Yeah, that's a great point.
but yeah, and now we're looking at we can spot out some of our favorite bacteria, Streptomyces, which we've talked about a little bit. And admittedly, lots of paper.
Oh yeah, number one, streptomyces. Well, strange, right? It's enriched in the plate sample here.
That wouldn't surprise me because actually, we know about it because we've been able to grow it from plates, and so I feel like a lot of the
Yeah.
bacteria that we know a lot about will recognize and notice that will grow from plates, but the ones we know very little about will be in the droplets. So
Yeah, no, that's a great point. Yeah, like more likely to recognize the things that are done in purple than in green. And actually, that's true for me. Like I know Pentabacillus, strangely enough, it's enriched in the
mm,
plates. It makes funny like patterns. It has really, and that's all about agar
yeah,
too. It makes really like funny patterns on agar.
yeah,
Okay, let's plow through. So they do it at every
mm,
level. I don't we don't have
yeah,
to talk about this, right? Like again, this is one of those things where as we know more about bacteria, right, this becomes a much more interesting discussion because we can say like, oh, I know that thing. Why do I think it would be enriched in this version versus the
mm,
other one, right? You can start saying things, but like that's kind of outside the scope of what this paper wants to talk about anyways. So just to say there are differences between, and they kind of hold out through all these different taxonomic levels.
yeah. So yeah, we're still going through these lists of wait a minute. Oh no, I was
Oh, you're
clicking the wrong button. Sorry, I was clicking. Sorry, yeah. Okay, so now we've bring us to figure S36, which summarizes a lot of that by looking at
yeah.
like the taxa that are exclusive to each section. So
Yeah. Focusing now on those that Venn diagram,
hmm,
right, and exploding out just the things that are in the unique sections of the Venn
hmm,
diagram. Let's rerun the diversity metrics on that and see, right? And you can see that exclusively to the plate, and we saw this already, right, in the broad phylum overview, right? Firmicutes. Firmicutes are the unique things that love to grow on plates. We know a lot of
hmm,
firmicutes, and that's because they grow on plates, and we're still seeing that here. But what's fun about the droplets is that they enrich for actinidobacteria, right? Which is that group that makes a lot of antibiotics.
yes.
So we're feeling good about the technique that they're deploying because it's, as you said, enriching the diversity in a way that we want.
And we're seeing things that we'd never really see on plates usually, like chloroflexi
Mhm.
or like planktomycetes. They're quite a
Yeah.
lot. So that'd be quite interesting. And also uncultured taxa, which is very interesting. Um, so
So when you
so,
when you take these groups and you just, I think they have a
hmm.
database of things that haven't been cultured before, and that's how they grab this uncultured number.
Yeah, I mean it's quite interesting the shared taxa, but I think the important thing to remember is there's very little here, and when you're sampling the soil, you're bound to find stuff that you've never seen before, just because there is so much diversity in the soil.
Yes, I mean, like, I think that I think that that is a great lesson for people again to say how diverse is stuff out there, even when you just go randomly into the soil and plate like hundreds. Remember, they did hundreds of
Hmm.
plates, so like this is not just like you go in, you do one plate, and you found something brand spanking new, right? You have to do a lot of replicates, but if you do a lot of replicates, you can find eight things that have never been found before, and chances are, if you did it again, you'd find those eight things again.
Yeah.
Okay.
So
What do we have?
next we have looking after growth. So taking those little microcolonies and then turning them into full colonies. So they had a neat little machine that drops them onto a plate as it rotates in a spiral.
Yeah, I love it. So like this is a way to bridge. So like scientists want to work with the
Hmm.
microbes that they find, right? And like one of the things that is difficult in flow, I mean, we've seen, I don't know if we've talked about cell sorting in flow, but you can sort cells in flow. It's just you only get like one or two cells around, and then you have to like fish them out of a tube, right? And you have to put those tubes on somewhere to isolate single cells, right? It's like it's not an easy process. So here they've yeah attached a motor basically to a XY stage to
Hmm.
the plate, so they can move the plate around, and then from the end of the tube comes out these droplets one by one, and they vary the speed of it so that they get single droplets all in a giant spiral around the plate. And you can see in C they have a because the oil evaporates. It's some weird type of oil.
Yeah, they got a liquid and a gaseous phase, and depending, because it's sort of like the way the like ethanol can kind of evaporate, or I feel like it's like that.
Yeah,
Um,
yeah, it's like that. It's but it leaves a film.
yeah,
And you see that film is the path, right? That
yeah.
like this spiral arm did. And along that path, they get to fit all these colonies in. Oh, I didn't put the number down. Do you remember how many they fit on a plate?
I do not remember how many they fit on a plate. But I remember.
I think it's a,
Sorry.
I think it's in the hundreds.
Yeah, it wouldn't surprise me that it'd be because I remember like I had to do similar things when I was like checking for mutants, and you could fit
Mm.
like I think I got about maybe like 30 or 40 on a plate by just puncturing like a pipette tip into
Oh,
areas.
by hand.
Yes, by hand, just by hand and by eye.
Yes, hundreds is not out of the
Yeah,
question, right? So you fit 100 droplets here.
yeah. I mean, I feel like a machine can be very precise in that, and it also depends on how long you let it grow, because that's the main limiting factor for how much you can fit on a plate. Because if you grow it for like maybe like an hour or so and pull it out, you'll have lots of microcolonies that are invisible to the eye, but are still growing and viable. And so it,
But don't remember. I think you have to remember here that these bacteria are being selected for slow growth in some ways,
yes,
right? Because they're getting 20 degrees for 28 days, and you can see in D they let the plate grow out to get the single colonies. That agar has dried out a
yeah,
lot. So like it takes a lot. These are slow growing bacteria, right? And they're giving them all the time they can because they want to find the rare ones.
yes. And you can see some parts of the plate where the bacteria have actually overgrown and start to flow into each other.
Yeah,
But that's
yeah. So that's the danger in this method.
yeah,
They could fix it, right? They could adjust it so that the spacing between the spiral is a bit bigger, and maybe like add dilute it a bit more, add more oil into the droplets so that like it takes longer for them. Yeah, move the plate faster and take more oil until a droplet comes out. So
yeah.
yeah, there'll be ways to
I
adjust
mean,
this if they need.
yeah. Even like sticking a camera over the top of them and like so it lets off an alarm, sets an alarm off, going, "Oh look, this colony is now this big. Just cut out that piece of agar and put it on a different plate." And then
Yeah, that's right. That's a good point. Okay, so they have this setup now that they can attach to their droplets, and they can sequence the colonies that grow from those droplets, right? Because the question well, no, please move to the
yes,
next slide. I
sorry,
think it's
yes,
good. The question is are the things that grow in the droplets because they're essentially using a plate again.
yeah,
Right? So like they have the potential to basically ruin all the work that they already did getting all these cool different things in droplets, and so they sequence them just to see what happened. And of course bacillus is like the most abundant
yeah,
uh, uh, coming out from here. So there is some sort of bias from plates, and they are losing right, a bit of their diversity by going down this method. But they still get some very strange things. Particularly, they get this uncultured cytophage
yes,
and so like that's kind of weird,
yeah.
right? Like this has never been cultured before, right? They grow it in droplets. The droplet hits a plate. It grows a colony on the plate, but they also note that when they restreak that colony again, it doesn't grow.
Yeah, I think
So
it's the
yeah.
actual like plating process is very important. I think that's a big like kind of filtering step because the way they did it in droplets,
Hmm.
it's almost all in liquid culture, and they let it like grow into a small like microculture, a
Yeah.
microcolony, and then drop it onto a plate in a specific way. And it's uh, I mean, admittedly, so there is always a question of whether you're seeing what you think you're seeing or whether you've just made a mistake. But sometimes weird things like that happen where like the stars align and something impossible happens and you're like, oh, this is amazing, let's repeat it, and it just doesn't repeat. And the question is, do we publish on this or do we not?
Well, it's also this is actually I think a growing body of literature talking about like what does it mean for some bacteria can spontaneously adapt to the plate condition,
Mm,
right? And like we see this with like lab strains that get passaged over and over again, they
mm,
change, right? Like living in a lab in freezers on plates changes a bacteria, right? And I think some people have even gone and said that this process is kind of domestication.
domestication. Yes,
Which
it is.
I just find a hilarious word to deploy in the world of bacteria.
But
Bacteria.
I mean, it's true. You have domesticated like various bacteria to make beer, cheeses, and various other
Hmm.
like things for us.
Hmm.
And yeah, I do feel like some bacteria have domesticated themselves by adapting to the lab, and some we have domesticated to create genes for us. There's like various strains of E. coli that are used to create proteins and other things. So they are very much. Some of them are have been adapted tools, but I think it's. I mean, listen, the interesting thing here is like the whole like there is still a filtering process that goes on just by things being able to survive or not survive on agar. That I think is a an
Yeah.
important story to hear, and it kind of also like it almost provides more kind of evidence for firmicutes and like gram-positive bacteria why are they more adaptable to agar than other bacteria.
Yeah, and maybe highlights an important bias that we have to think about, right? When you design your bacterial study to look at something, right? And one of the steps is, oh, I'm going to plate them on agar and pick what grows, right? Like you're already adding this filter for yourself. And so anyways, so they're able to use this technique. The droplets do grow unique things. They can put them onto plates so we can recover them afterwards, but they lose some of that diversity, right, by putting it on a plate.
Mm,
The authors do point out that there's still these prominent isolates from actinobacteria,
mm.
right, which is things that probably they say maybe not would have grown on the plates originally, right? So at least I think this goes back to the point they're enriching for some sort of diversity by passing it through the droplet cultivation method.
Right. Yeah, and I think the next step is where things get more exciting because this is where they start using droplets to screen for antibiotics. So let's see if I can get this video to play.
Oh no, it looks
Uh
dark.
oh. Okay.
Oh,
Hold on a minute. Okay. Right. Okay. So the video doesn't appear to be. Okay. Video isn't working unfortunately. So let's see if I can. Uh,
we stared at this video in the last episode for a while too.
Yeah, we did stare at this video for a while. So let's see if I can get. So I'll pull up see if I can pull up the original paper quickly. Uh, uh,
So while you're pulling it up, I'll explain, right? So again, the authors have already done this work before, right, in terms of piloting this method, but now they have a cool video to show us. And essentially, what that work amounts to is once they have their droplets that have their colony of interest growing inside of it, so that's after the 28 days, right? So they've spent 28 days sitting in a liquid, growing up, getting more biomass. So now they're going to put those droplets back into like some sort of microfluidic chamber, and as the droplets pass by, essentially a spigot that is filled with another culture of bacteria. This time, a fluorescent reporter strain, right? There's a short zap of electricity. I don't understand how these things work. This is the microfluidics world, but I guess in the microfluidics world, you can use electricity to disrupt surface tension to let bubbles attach together. And so as these uh, bubbles pass through the spigot, the short zap of electricity is used to put a droplet of that reporter culture into every droplet that passes.
so I think I've got the video showing now. Uh,
Yeah, I can see it. Looks good.
let's see. Okay, right, excellent. Okay, so yeah, this actually
Right?
shows what you've just been explaining.
Mm-hmm. Yeah, so remember, like, this is pretty cool because there are 9 times 10 to the 6, is that how they're doing it? Droplets in this mixture. So that's a lot of droplets to go through, and they just have to flow them through this device, and now every droplet gets its own little bit of reporter
Mm,
strain. Um, But once again, this is going to introduce a bit more noise into our scenario because in addition to getting the reporter strain, it also has media that the reporter strain needs to grow in.
Mm.
And so there could be other effects going on where like that media, because like the bacteria that we're screening, they don't grow in that media, right? We know that they don't grow in that media because they've been growing in cold extracted soil, right? Cold brew soil all this time. And so like, there's another little complication here, but again, previously published that they were able to screen through for antibiotic production in these circumstances. So let's see so they go and they do it.
Yeah. And so the next stage of their screen is where they sort the droplets into ones which have the reporter and which ones don't.
Yes.
Um,
So just importantly to remember, they wait again 28 days at 20 degrees,
mm,
right? So they basically give the bacteria time to grow again, both the reporter strain and the strain that we don't know. Of course, we don't know what's going to happen with the strain, the unknown strain, because it might not grow in that media. But hopefully, if all goes well, right, the reporter strain will be suppressed in growth in some ways. And the ones that are suppressed in growth, they're fluorescent, so those droplets will not fluoresce. And they use a little bit of electricity again
yep.
to sort them. So I think probably when the droplet first comes out, it gets shot by a laser. You can't really see any of this, right? Because it's all out of the plane. But the
Yeah.
droplet first gets shot by a laser. If it glows, then it goes into one lane. If it doesn't glow, it goes into the other lane.
Yeah. So we can.
Mesmerizing. I
Yeah.
love
It
it.
is. Yeah. It's excellent. I'm gonna have to put it back to our slides in a bit, so I just transition us while I
Okay.
do some tech stuff in the background.
Yeah. Okay. So after that sorting happens, they take the results of the ones that have suppressed and they put that on a plate, right? So that's why they out this plate technique because at the end of the day we want to recover the bacteria, right, that we have screened that was able to suppress that growth. And um, and and we want to know, and you have to kind of do follow-up studies now, right, because you have the bacteria on a plate and you have to make sure, was that suppression in fluorescence because of what was growing there or for some other reason.
Right.
Okay, but in figure six, they're just going to give us an overview of the diversity again that's coming off of the plate. And you can see that media has a huge effect here,
Yeah.
right? Like the B subtlest in the maltose, I guess it's the
Yeah.
maltose.
Maltose
Yeah,
takes
that
over completely.
it takes over completely in the sense that I guess a combination of having that bacteria and that media together means that only other bacillus will grow.
Yes.
So you still get hits, right? Like
Mm.
it's not that there are no hits in this condition, right? Like you still get, oh yeah, it suppressed the growth of this bacillus, it suppressed the growth of this bacillus, but the only things that you get that are hits there are other bacillus, right? Because again, they've been manipulated too much now.
And
Uh,
another thing is that these are they list us the bacteria that they tested on. So that when they mixed bacillus with other bacillus, when they gave a reporter strain that was B subtilis, it was more likely to be killed by other bacillus, which is
mhm.
part of like this thing of bacteriocins where bacillus are constantly fighting each other with like microbag with other like kind of
Yeah.
chemicals.
Uh-huh. No, I mean it's kind of interesting from that sense. I mean these are really interesting observations actually that in of themselves could be used to support some hypotheses, right, about the natural world. But
Mm.
in this context, they're very much like bugs in the system.
Yeah.
But they're like, darn, like we didn't want to find more bacillus that fight other bacillus. We want to find like other things that we can use. That being said, without doing follow-ups on these different things, these are just screening results,
Mm.
right? And screening, there could be many reasons why these results happen. Some of them are not even antibiotic related, right? Like some of this could be contact, right? Sort of
Yeah.
killing that happens between the bacteria. So they do a primary, oh, we'll show it later, but they do primary validation just to make sure that when they follow up on these things, it's not due to some of the other effects that could happen in the drop.
Right. So that brings us to figure S. So this is where they follow up on that cytophagaceae, uh, story
Yeah,
with
so
the
basically they find it again when they did this experiment.
right.
But when they align those sequences to the one that they found before, it's a different one.
Mm,
So actually, I love this B plot to the paper because it really like excites me about that whole uncultured microbiome story, right? That like, does that mean they've accidentally stumbled upon a technique that can enrich for this particular genus of bacteria? And if so, why?
yeah, I mean, did they do something new, or was everyone else just like messing up before this? Like, we're gonna
Yeah,
culture this on fire. Oh, nothing grew.
yeah.
But maybe it's just this extracted soil medium that's working.
They talk about the story they talk about again is that on the plates after they spot the drops they do get it to grow, but then when they try to restreak it they can't get it to grow anymore. So like there is a story here about this stuff does not want to grow on plates. Their method definitely allows this thing to grow in droplet,
Mm,
but you know how, why, what's the difference, right? And like of course this is like a whole other paper's worth of things, and I think actually the reviewers do the reviewers of this paper do fixate it on a little bit, not like to
mm,
point it out specifically, but the authors talk a lot about the difference between droplet and solid, but they never really show like what that difference might be like generated by or like verify, right? Like
mm.
that the mechanism is the same when they initially played it versus after they droplet. To me, it's fine because that's not really the point of the paper, right? The point of the paper is here's a cool technique to find antibiotics, but it does, in my mind, elevate like very interesting thoughts about like what does determine, right, those like that one thing would grow in one environment and another. um and if you think
Yeah.
soil, it's an incredibly complex environment, right? So many different things are going on. So like so many microenvironments exist. And this whole droplet technique is interesting because you basically get to target a type of microenvironment. What type, I don't know, but it is something, and it seems to at least be more amenable to certain types of growth than others.
Well, I mean, I think the really interesting thing with the droplets is that it allows you to do liquid culture, but sampling with it. So, because usually when you take a sample of liquid culture, one strain of bacteria will just take over completely because there is no kind of way to stop it, no way to protect other bacteria. So if you enrich it, the bacteria that. Whereas this droplet culture allows you to do the liquid cultures, but in a way that preserves that diversity, which is quite
Yes, yes,
interesting.
absolutely. And so, but see, that's why I'm saying that like that statement that you made is like very much about the difference between liquid and solid culture. That there's some advantage to using liquid culture in this method. And one of the reviewers pointed out it wasn't addressed, right? Is that like then show us the microplate experiments, right?
True.
Then repeat everything, right? I mean, of course, like you don't want to force them to do a million microplates, right? But do a subsample, right? Do like 1,000 microplate wells, right? And then subsample the droplets at 1,000 and show us the difference, right? I think that that's something that they could have addressed, right? If that was the claim of the paper, but I don't think
Mm.
that's the claim of the paper, right? Like I think that they are more interested in just like the technology as it exists, not like how it stacks up to other things.
Right.
Yeah. Anyways, I think it's cool. I love the B plot. I think there's a lot of questions I could spin off of it, but I can see that the focus of this lab is actually drug discovery. So
Yes.
that's not what they're going for.
Speaking of drug discovery, they actually found hits and they decided to validate those hits to see what exactly what did they find a new antibiotic? Um,
Yes. I think it's really important to highlight the way that they validated it as well. They did these validations by growing them in a whole bunch of different medias because they didn't
mm,
know which media would be the best for making the particular compounds that they want.
mm,
And then they filtered those medias so to remove that potential effect that like it's the bacteria itself is like contacting the other bacteria and killing it. Because that happens in the bacterial
yes.
world. We should definitely talk about that at some point in the future, right? All this contact dependent killing. Um, so yeah, they do this, and this is their primary validation, and they validate against a whole slew of different bacteria. So
Mm,
you can see in table S2, they look at Bacillus subtilis, Staph aureus, E. coli, Pseudomonas, Mycobacterium, Sporobolomycetes, Candida albicans. That's a pathogenic yeast, and
yep.
penicillin, which is yeah. type of fungi.
Yeah.
The Candida and Penicillin are both fungi. So they do a whole slew of those with all these things, and they have one hit that they want to show us. It's a bacillus isolate that they never rename, so it's D1210906B321, which was kind of cool to them. I don't really understand this. In D, they show that like it grows when it gets exposed to M. vaccae.
Yeah.
Don't really understand what they're trying to tell us here. It's very confusing to me. I feel like they didn't pay a lot of attention to this follow up. Um, but they are highlighting something interesting is that they did primary validation. They do get hits.
Yes.
Um, they also did a whole bunch of different medias. The table S5 excerpt that I picked out for us here only is to explain the difference between A and B
Mm.
on the primary screen that they show, but they actually have like 9 different medias in their compositions. So the fact to me, this is like one of the things, the reading between the lines part. I mean, you could just look up the lab and see what it does, but like when I see so much methodology kind of hidden away underneath it, it really shows me that like this paper, the lab is set up to do a very specific type of thing. They're very comfortable doing a specific type of thing, and so like the whole microbiology part of this paper is almost just a side part of their studies, right? And their main studies is actually the work that's packed into the last parts of this figure,
Mm.
which basically don't really get supported very well. But it's actually, to me, the focus of this lab is the way I understand it.
Yeah. Uh, so let's bring on to Figure 7A, where they try to look at the different kind of molecules that they find, I guess.
Yeah, so it's one thing to say that the things that the bacteria make inhibit the growth, but you want to drive down in drug discovery, you want to say, but what's the molecule that does it,
Yeah.
right? So there's a, we haven't really talked too much about this technique, but there's a technique called liquid chromatography where you pass a complex mixture of things over a standard sort of slightly sticky material, right? In this case, they're using C18, which selects for hydrophobicity. And so they pass it through this slightly sticky thing. They elute using another solvent at different concentrations, right? At increasing concentrations, the substances that stick or don't stick to that column. And in that way, they're able to separate out all of the different things that are inside of a mixture. And then you can take those fractions, you can test them. In this case, they test them using
Yeah.
spectrometry, meaning that they find the masses of the molecules that fall off the column. And then you can do some also crazy big data techniques to compare all the known masses of things that exist out there against the masses you find in order to try to get an idea of what's in there. So that's what they're doing in A. You can see they find 5 different peaks that they're interested in. So they're going to follow up on that. And from top to bottom, those are all the different techniques that they use to grow up the bacteria and then to extract the compounds out. So this is chemistry in some ways,
Yeah.
right? Like some very heavy chemistry that they're doing to find in this complex mixture that a bacteria makes, what is the molecule that's doing the work?
Right. And I think they
And
yeah.
on the left hand or on the right hand side, that is a very similar thing. It's an elution. They're just not doing mass spectrometry here. They're doing some different types of. Oh, I think they are. Maybe TIC is the type of mass spectrometry, but they're also doing some other types of analysis here to look at what's in the samples.
Right. And what they find is they find this molecule called a bacilline.
Bacilla,
Bas
yeah, Bacilla Ains.
yeah.
Oh,
But
Bacilla Eads.
because it's bacillines, but that A is throwing me off because
Yeah.
they just put an A in there, just feel like yes, but yeah, but so the
Yeah,
bacilline
so
A and
this
bacilline B.
molecule already known, right? They actually they know it because the mass spectrometry is extremely precise,
Mm,
right? It gives you like down to the it actually gives you down to the ionization state.
yes,
Like what the mass of something is. So like the salt of something is not the same as the mass of the ion dissolved in a liquid. And so from that very precise mass, they actually they already build the hypothesis that, oh, it's peaks 1 and 2, they are the bacillines. And they double check that using UV spectroscopy. And then they also do a bunch of like tandem mass spec. See, this is where I'm like, they did not show enough figures to really give us this understanding, because no way they're expecting to like believe just by looking at these mass, like no one has these mass specs memorized.
Yeah, true.
So like just by showing us the tandem mass specs, like no, you have to show me the comparison, or you have to show me the standard, and like, or like highlight the peak or something that tells me like what's going on, because I cannot for the life of me read what's going on here.
Yeah.
You just gotta trust the authors.
Yeah, I have no. I mean, we might have a chemist in our audience who might be able to look at these peaks and tell us like how messy these samples are or what kind of things were happening. But yeah,
More than that, they say that they say that in one of these mass graphs, I think it's like C or something like that. One of these, I don't remember.
mm,
I'm not going to go through. They say that the shift that they see there, right, is after tandem. So after things got blown apart, they basically shoot ions at these pieces that come off the mass spec, and then they blow up into even more parts, and they find the mass of those parts,
yeah,
right? And based on like the types of ions that they shoot in, they know that certain bonds break, right? And they can see like based on the resultant masses what that bond might have been, right? And what's on either side of that bond as
yeah.
well. And like you can't just show a single mass spec and say that that's what it is, right? Like there's a level of trust going on here for sure. But
Yeah, I feel like they're taking advantage of the fact that these are known molecules, so we're gonna take them on trust. If it was something completely new, I feel there'd be a lot more scrutiny into this.
yes. Yes. Yeah, they're not showing us super anything novel here. In some ways, this is just these are the test results that come back from the lab that then say, da-da, this is what it
Mm.
is. Right. So they've given us the test results. But yeah, just from the reading the paper perspective, I found this part a little bit frustrating, because I was like, yeah, it's kind of nice to see the data,
Yeah,
right? To know how it was done. And like, yeah, mass spec things just go so far. Anyways, already known. They found a molecule that's already known.
yeah. So
Which maybe isn't too surprising, because they're looking at a bacillus, right? We know a lot of stuff from bacillus.
yeah, I mean the way their filtering process it produces a lot of diversity, but it's not necessarily going. I mean, this is almost a proof of concept, so that they go, oh look, we found something that we already know works. Therefore, if we find something else that works, and maybe, I, yeah, I can imagine it's a bit disappointing that they wanted to, because it would really push this paper that much further if they found something completely new. Um,
Yeah, can it be proof of concept that gives us something we've never seen before, right? Like that's the story. That's why I sort of led with the eye chip
yeah,
technology, right? Because this is supposed to be positioning itself as a way of getting to that. But actually, upon reading, I'm not sure if it does, but I think there are other interesting things that it can show.
yeah.
But again, follow up on that particular line of reasoning.
Yeah, I think that's something that I'd like to follow up on. They also found surfactants
Yeah.
as well that
Yeah.
were also.
So this one, they didn't do this one by mass. They did this one by retention time. They were like,
Mm,
oh my God, this stuck on the column for so long. There's not many things that stick on columns for that long. So they
mm.
ran a standard of something they thought would be the thing it is, right? A surfactant, because we know Bacillus makes surfactants. We talked about it in our swarming video.
Yes, we did.
um, and we talked about surfactants as like enabling mobility, but they're also a type of antimicrobial, because they're kind of lipophilic. They can like eat up membranes and stuff. So then in this one, they ran a standard surfactant. They're like, yep, it's the same thing. It comes off at the exact same time off the column. And then once again, they dive into the mass spec, and you got to trust them again that the mass spec shows that it is this
Yeah,
surfactant
I'm
that
going
they're
cross-eyed looking at these mass specs. But yeah,
Yeah. So all in all, they've done this primary screen. They've fractionated stuff. They found 5 compounds of interest. 2 of them are known strong antibiotics. 2 of them are lesser known antibiotics, but still known. They end up rerunning their fractions in an inhibition assay, just to sort of get a sense of like which one is doing the inhibition, right? Like
mm,
they know that when they put the entire lysate in, or not lysate, the entire supernatant in, they get a zone of inhibition, but they are positioned to try to figure out which one does it more. And so they fractionate it over liquid chromatography, and they place them into all these different wells. The fraction that has the bacillines,
Yep.
which is D, that has the largest zone of inhibition compared to the ones that correspond with surfactants, G and H. Um,
Yeah.
yeah, G to H, so G, FH. And yeah, and Cipro has a huge.
Yeah,
That's
ciprofloxacin, it works, so that kind of shows up all the rest of the things they found. But
yeah. So yeah, and that's what they leave us. This paper leaves us with. They've extended their previous work. I think is the biggest thing that they've shown in this paper. Right? They've extended their previous work not just to work on a toy example
mm,
where they know the type of bacteria that they're putting in the droplets, but they now have a method where they can take a community of things and put them into droplets. And furthermore, they have this really weird indication that, at least for one round of plating, they're able to capture some strange bacteria that they wouldn't have been able to capture otherwise. And yeah, and maybe that could help us find antibiotics, but I don't know.
yeah, this is an interesting one because I feel like it's not so much capturing, it's selecting a different area of diversity that hasn't been covered previously in microbiology.
Yeah,
Uh, but
that's
there are but there are still filters in there that are preventing things that could be interesting from growing. So we're still coming
uh,
up against the same problem. We're just like lessening it somewhat. It's not so much.
yeah, like it. Uh, what you said before, which I think is a really awesome thought as well, right? Like maybe this is just the idea of doing things in millions of test tubes, right?
Mm,
Instead of millions of plates. But like in some ways, we've already brute forced our way through the plates problem,
mm,
right? Because if it really is just if this technique all it is just plates, then robots already do plates,
yeah.
right? And like a robot working for some time, like maybe this shaves off some time, right? But like if we have to go back, if we still have to ultimately go back to, if you have to grow up these samples on the plate, then it doesn't really fully solve the problem, I
Mm,
guess, right? And actually, I find myself at this present moment after reading this paper and talking out with you and hearing that plate thing again and thinking about the review from the reviewers, I do feel like they should have done the microplate experiment, because it would have at least shown us more of the utility of this method itself,
mm.
right? I want to know the utility of the method itself, and it could be though that it's already done, right? As I said, like this is not like this is a very active line of research, right? Like in 2020, right? Like people are growing lots of things in this format, and so it could be that like that's just not what these people. This is not what these authors are interested in. These authors do want to find antibiotics. They are using this method. This is their next step in that chain, right? It didn't find anything yet, but they still have many more hits to follow up on, and perhaps with tweaking, this method could be used for different things or could be even better for finding bacteria.
Yeah, and I guess I want to go back to the extracted soil samples because the soil process seems to be quite intense, and they test that
Yes.
using DNA. But DNA doesn't always get destroyed when a cell gets destroyed. So the
Yeah.
question is whether those bacteria they sequence from the samples were actually viable or not. I don't think I feel like they will answer that question. Uh,
Oh, interesting. The singleton reads, right? Like,
yeah,
what if it's diluted out like into the droplets a little bit of broken DNA,
yeah.
right? And then that ended up getting picked up, and then that's what we're seeing.
But I mean, there's a lot here because I feel like the droplet methods there is definitely some interesting things that could be done with them. I mean, in terms of like scaling up the way we do the scaling up testing for antibiotic
Well,
resist.
one of the ways they could have gotten around that I didn't read in the methods is they could have only sequenced from the things that were positive on a optical microscope, right?
Right.
Because like they can see the stuff that grows. Random fragments of DNA will not be those things,
Mm.
and so if they when they did their sequencing, we can just look at this right away. When they did their sequencing, if they did a step where they only sequenced the droplets with stuff in it after the 28 days, that would give us that would give us something, right? The other thing is though, it's pretty rare if it was just remnant. Actually, I'm not sure. I can't speak to that. I'm not sure how like library prep stuff,
Yeah.
if it would have amplified something like that.
Oh, I mean, I feel like they with if when they're doing their genomics, it would have been quite interesting if they did some barcoding with that. So rather than just mixing everything together with metagenomics, like just adding in individual barcodes so we could tell whether genes were in the same thing or not. I thought that would have been quite an interesting thing to do.
Oh, sure. Yeah. I mean, that's actually they can add that to their methodology, right? You can add a barcode. You can add a beaded, sorry, a barcoded bead into every droplet, right? So
Yeah.
that when you run, and then you can run your library prep in that emulsion. I don't think they do that. They don't run their
No.
library prep emulsion. They just mix it all together and do it.
Yeah, but again, you're right. They weren't focusing on. They were very focused on finding antibiotics. Um,
Yes, exactly. Yeah, so it's kind of like I'm glad the paper got published, right? Because it's good that scientists are keeping us abreast of their work, right? And I think that they don't claim anything too crazy, right? At the end of the day, reviewers are also just making sure that the claims match up, right, with the data. And if the results don't claim that much, then it's not a big deal, right? Like we don't have to like rip it apart. We don't have to rip it to shreds because there's still the things that they've shown, the data that they've shown that we're learning from.
yeah,
Um,
I mean, what they have is a good like kind of baseline that can be built on for further developments. I mean, we've got like this
yep.
quite interesting system. We've got a way of, I mean, as you said, one way to extend this would be to use those microplate methods to try and uh, look at the questions that way.
Plates, and then I mean, you suggested beads. That's also a good way. Yeah, I'm reading DNA extraction purification. It doesn't sound like they do anything to sort their droplets before they do the purification.
Hmm.
So I think a sorting method would be really cool to kind of it would strengthen their diversity claims,
Yes.
right? Then again, there may be a good reason, like from the library prep perspective, as to why that is an unnecessary experiment. Because it could be that like a remnant DNA in that mode would be diluted down to such an amount, right, that it
Hmm.
wouldn't be picked up by the reads. Because you got to remember, like they do for the bacteria that they are able to sequence in droplets. There's a small colony of them, right? So that means there's a lot more DNA from that than from like a single bacteria that got like ripped to shreds.
Yeah, and another thing that like the thing is in soil that the bacteria are often like interacting with each other. You get syntrophs, and I think that this would be an interesting tool for looking at that, where you actually take these droplets, separate them out, barcode them, and then mix them together to see whether there's any improvement to growth. Uh,
Yeah.
and
Well, I mean, this is the first time I've really thought about microfluidics in this realm. I
yeah,
mean, I got obviously I found a review on it, so like people have been thinking about it, but like that just sounds cool. Like separate things out singly, right? And then using that same technique they use for injection, but instead just fusing two droplets together.
yeah, that would
So
be
you do the test where you grow them all separately, right? You grow them all up, and now they're all like in like these tiny tubes, and then you pairwise fuse them together.
yeah.
And if you do a lot of it, you might find combinations that you didn't think about before, right? You're like, oh, we sequenced this droplet, it had two bacteria in it. We never thought they worked together, but they grew to a really high amount.
And that has more because I remember in our news shows we covered like some papers looking at syntrophs like for creating methane or creating
Mhm.
biofuels. And again, this
Yep.
is the kind of thing that because understanding how the microbiome like works within itself is important in itself because it can not just affect antibiotics, it can affect like things like biofuels. It can affect things
Yep.
like developing anywhere where we use bacteria to do something. The
To- totally.
so it's an exciting new method for bioprospecting, and sometimes the value of a paper isn't so much the fact that it tells you something new. It kind of introduces a new technology to a new field of researchers who can use it. And
Yeah, absolutely. And it documents the work that was done,
yes,
right? I think that's a really important thing. Like we're seeing a documentation of work here, right? Like um, we can judge independently how valuable we think that work was,
yeah,
right? In of itself, but like even just seeing that documentation means that we could pick up these tools. We can contact these people, right? I mean, if we were researchers in a lab or if we had access to the right supplies, we could contact and follow up on some of these results. um, yeah, I think that it's it's an interesting paper in that it has got me thinking about microfluidics. I do think that there is some promise in terms of screening for
Mm.
antibiotics in here, but I already see like a bunch of technical limitations, right? And there has to be a lot more sort of engineering work, I think, and like really thought onto how to remove those limitations. Like that reporter strain, right? It has to come with its own media,
Yeah.
right? That the only way to isolate things would be to drop them on a plate. Oh, like you can drop things into micro wells,
Yeah.
right? You don't have, if that's really such a big thing, you could maybe adjust it so it gets dropped into a micro well instead of on a plate. It looked like they were already using a type of software to like see the droplet coming down the pipe.
Yeah.
They had like a microscope there, so then they knew the time the droplet would come out approximately, and so you could try to time it out so it fell into different plate wells. That could be a really interesting way to go ahead and do it. Yeah.
Yeah. I mean, I do feel like this is like one of those papers where it's like kind of where it's like watching a bad movie where you're like, oh god, there's so much more they could have done with that without appreciating what it is. So
Yeah.
it's like that where this is like sparked off a lot of like imagination for me. Like I quite like
Yeah, yeah, but in terms of where it actually fits, I think it's just a stepping stone in this lab's quest to optimize this technology, right? Like I wouldn't be surprised if some of the things we said they've tried, right, and it doesn't work for various reasons, right? They've been thinking about it and they've still hit a wall. Like maybe they go to a couple more conferences, they talk to more of their colleagues, right, and then this idea would develop more.
yeah, and I'm quite interested in this because this is very much in the lab on a chip technology kind of thing that, and there's a lot of I mean, it's amazing like how little we've seen the like engineering like world of miniaturization impact microbiology until very recently, and it'd be
Yes,
quite interesting to see how that like progresses for the future.
for sure. Anyways, that's I think that's everything I want to say about this paper. It was really interesting, and yeah, how do you feel? Ready to move?
Yeah, I'm ready to move on from this paper. I think there's a lot of really interesting stuff here. Um, and like yeah,
Yeah. If you enjoyed that paper, you should join us next week for a news week where we're going to survey papers to find something that we want to cover in detail the following week. And I think we're also going to follow up on one of our viewer comments next week and try to maybe speak about SARS-CoV-2 reservoirs.
yes, like I guess there might be some controversy in the field that we're going to try and delve into, so that'll be interesting. Um,
Yep.
and we want to remind everyone that while we're very enthusiastic about microbiology and somewhat qualified, it's possible we didn't get everything right here. Science is about thinking critically and asking the right questions. So if you have any questions or corrections, please let us know in the comments.
I totally agree. You can reach out to us over Twitter with the hashtag #MicroTWJC. We both believe that peer review is a process that we can all participate in. So we hope that you've had a good time listening to us ramble about microbiology today. And if you think you have something to add or you found something unclear, just let us know in the comments.
It's been a pleasure chatting with you, Danny.
Same here, Paz.
Tune in next week for more microbiology content.